Good morning. So today I'm going to try to convey to you something that I'm very passionate about, which is sort of leveraging my patient population as a veterinarian to facilitate drug and device development in both human medicine and veterinary medicine. And it's something that I sort of fell into years ago, and as a sort of pseudo specialty, it's grown tremendously over the past 20 years. So Dr. Jackson alluded to this earlier, which is that when we think about drug and device development in the traditional sense, it's been very linear, where you have this sort of this incubator that occurs in the preclinical realm that takes anywhere from 3 to 6 years, where you may identify a target, you develop validation assays, you may put it into some mouse models, and then you do your GLP testing. And then you eventually get into people, and that's where the drag often occurs. And it can take 10 or more years moving from your initial application to phase 1, 2, 3 studies to a new drug application that's successful. So that's an enormously long process. And if you fail all the way up here, that's up to 20 years of time wasted on an effort that ultimately cost millions of dollars and failed. So when we think about how these processes typically occur, you have your realm of drug and device discovery, preclinical investigations, and then clinical trials. And I'm going to focus mainly on these preclinical investigations today. So again, Dr. Jackson alluded to this problem that we have in the drug development scheme, which is that the overwhelming majority of efforts fail once they get into the human arena. And so this is an older slide from 2014 where they looked at overall failures in the phase 2 setting and the phase 3 setting, and then the likelihood of approval. And you can see that for some entities, it's not as bad. You have a 17% likelihood of approval, but for other disease entities, it's really abysmal. 7% in cardiovascular disease, 9% in neurology, and then overall, if you average it, only 10% of drugs that sort of enter the human arena make it to a successful new drug application. That's like a 90% failure rate, which is enormous amounts of failure. And in any other setting, in any other business model, it would not be acceptable as a failure rate. So there are also some disease entities where there's really absolutely been no movement of the needle at all. And I think you are all probably familiar with some of these, and where billions of dollars have been spent and nothing really has happened. So there's several neurodegenerative diseases like ALS, where really nothing has changed. Alzheimer's disease, acute kidney injury, some bad cancers like glioblastoma and pancreatic cancer, we really haven't improved outcomes at all. Acute respiratory distress syndrome, and another disease called idiopathic pulmonary fibrosis. So these are just some of the examples where we really haven't improved substantially outcomes for patients affected by these diseases. So what does this mean in terms of an economy model? Well, when you fail that much, you're destined to really essentially be dumping money in a way that becomes essentially not sustainable, particularly when the cost for doing trials goes up. So if you look back to the 1950s, you had about 40 to 50 drugs approved per $1 billion spent. So that's not so bad. Now you get less than one drug approved per $1 billion spent. So enormous amounts of money and resources are going into efforts that ultimately failed. And this sort of money dump is not sustainable long term. When you look at the reasons why these failures occur, they're really in two broad categories. One is efficacy, meaning that you spent all this time and effort and then you put it into people and then the drug is just simply not effective. And then the second reason is something called safety, meaning that you did all this preclinical modeling, all this testing, and then there's a bad safety signal that occurs in patients that wasn't predicted based on the models that you were using. So basically, 80 to 90% of the reason for failure are the drug's not effective and it's not safe. And you think, okay, I just spent 15 years working on this. How can I not know this is not effective and not safe? And so this is the conundrum that we face, is that we think that we know what we're doing. We spend a lot of time and effort in various models, and then we have confidence going into a disease in humans based on data that we've accumulated over a period of time, and then that data ultimately turns out to be wrong. So there are many inefficiencies in the process of what we call pre-IND IDE process. Sometimes it's hard to find an appropriate drug device for development, meaning that some look really good early on but fail later in vivo due to unanticipated toxicities or off-target effects. So you have something in the laboratory that looks great. Everything is perfect. You have the right target. It's very specific, and then you put it in a patient or a mouse or another model, and then it has a toxicity that really makes it not an appropriate drug to move forward. One of the largest challenges that we face in this preclinical realm is identifying the best model to test efficacy. So which model is it a rodent model? Is it a genetically engineered model? Should we be using sheep, et cetera? So we really don't always know what the best models are to test effectiveness. And this is particularly relevant given the fact that if you look at the spectrum of human disease, it's not one thing. ALS presents as many different entities. There are many different subtypes. People present with comorbidities, meaning they're smokers or drinkers. They may be overweight. They may be too thin. And so they present to you with a disease, but they have all these other factors that play into it. When we think about the models that we use, they're typically not heterogeneous. We want our models to look exactly the same so we can get statistically significant data and keep the variables low. But that's really not the human condition. Sometimes it's challenging to develop methods to scale up or formulate a new product or a device. There could be significant technical hurdles. So things that work very well in a small scale setting may not translate to larger models. And then formulations in mice or smaller mammals may not be compatible with giving things to humans or larger animals. And with respect to device development, we're working with a company now that has a really novel drug delivery device, but they're facing challenges with respect to how to place that instrument. And the model systems they've been using are really not predictive. So we're working on trying to find them the right model that will help them really optimize that device before it goes into people. And so when we talk specifically about cancer, which is the field that I work in, there are many features of our classic model, the rodent model, that are not consistent with how cancer presents in either our pets or in people. So many of the mouse models that we use are immunodeficient. So they simply don't have an immune system so that they can host the human cells or cell lines. Many of the what we call patient-derived xenograft models, they're immunodeficient. So that really doesn't represent the landscape of human disease because we all have an immune system. The other thing that is really different in many of the rodent models of cancer is the accelerated timeline of development. So when you're doing rodent studies, you need to have them occur pretty quickly. Like you don't want to wait a year or two for a tumor to develop and then try an interventional study because it would take too long to accumulate that data. Plus, mice only live for about 1 to 2 years anyway. So you have a relatively short timeline that has to be compressed in order for you to generate your data. So that's not what happens in the normal scheme of cancer development. Tumors can take months to years to develop. And during that time, there's sort of iterative process with the microenvironment where the tumor cell is adjusting to where it's living, things are changing. You really force the timelines in the rodent models, and so what you get may not be the same as what would happen if this occurred over 10 years. Mouse cancers can be relatively homogeneous as opposed to human cancers, which are relatively heterogeneous. The other important thing that is very challenging to evaluate in mouse models is the tolerance to a therapeutic. Mice are very tolerant to things that you do to them. It's extremely difficult to evaluate chronic toxicities that can lead to a reduction in quality of life. So patient-related outcomes related to quality of life are really important on the human side, and that's just not something you can assess in the mouse model. When we think about how we assess toxicity in mice, we look at ruffled fur and weight loss. And so we can't really ask them, how do you feel today? Do you feel like getting up and going to work, sort of thing? So it's very challenging to look at quality of life endpoints in those models. And then again, as I mentioned before, mice don't have comorbidities like the human population does. So, and when we think about testing new therapies in people, we're typically testing those in patients who failed a regimen of other things. I was looking at a drug approval for one of the drugs that we'd work with, and on the label, it's approved for what we call pentarefractory multiple myeloma. That means that patient would have had to have failed 5 regimens before it can get this regimen. We just don't see that sort of modeling in mice. We're not creating these models that are multi-drug resistant and then trying to treat them because we really can't. They don't live that long. So how can we improve translational efficacies? So one is better predictive modeling using sophisticated informatics tools. So informatics is really playing a huge role now in predictive modeling. There's a lot of work, particularly in the biologics field, looking at how, for example, antibodies bind, how long they bind, where they bind, modeling the regions in which they bind. So we can use a lot of tools with respect to informatics to figure out how a therapeutic might work. And these are tools that weren't available to us, 5 to 10 years ago. One of probably the key things to do is to employ the correct model to answer a key question. And so if you're using a model that isn't going to answer the question in the correct sort of landscape that you need to be answered, then you'll get an answer, but it's not going to apply to the human condition. So for example, cardiac toxicity of anti-cancer agents is often studied in mice. So if you look at mice, their heart rate is 300 to 800 beats per minute. That's phenomenally fast. It's extremely challenging to image the mouse heart. People do it, you can do it, but it's a totally different landscape than it would be on the human side. They also have 80 to 230 breaths per minute, making dynamic evaluation very challenging. You really can't do the state-of-the-art imaging, which is cardiac gating in a mouse model. It's just not possible because their heart beats too fast and they're breathing too quickly. There are better models out there, but they're often not used because they're more expensive, more challenging. So there are mini pigs, which can be used for cardiotoxicity. And dogs, it turns out, get cardiac toxicity from many of the chemo agents and could be used as well. But if you look at the landscape of what's published, most of it's in the mouse model. So it's hard to draw direct lines between a mouse that has a heart that beats 300 to 800 beats per minute to a human who's much slower. There are now pushes to take these immunodeficient mice and make them humanized, meaning put a human immune system into them. And those may be more accurate in predicting efficacy of immunotherapy strategies. However, the immune landscape in a mouse human hybrid model is still not the same as a human because many of the components of the microenvironment are still mouse components. And there are often incompatibilities between cytokines and receptors that limit the ability of those immune cells to function sort of the same way they would in a human environment. And importantly, and again, something that Dr. Jackson alluded to, is that this really should be a team approach when we think about modeling. Like, you really need to think about models as not the answer or the way you sort of push something forward. There's not one model that's going to give you your answer. You need to grab information from multiple models and put those pieces together rather than relying on just one. And this is sort of an iterative approach that will likely improve your endpoint assessments like activity and toxicity. So you may be able to answer one question in a mouse, like, can we inhibit that drug target or that protein using this drug, but you may have to put it in another species to look at pharmacokinetics because it's very different in the mouse. So you may have to pick and choose which pieces of the puzzle you put together to really create sort of this comprehensive three-dimensional model that is more reflective of what you would see in people. And what I'm going to talk about today is how we've been able to incorporate spontaneous disease models in our veterinary patients into drug device evaluation. So if we look at spontaneous diseases in companion animals, there's a whole lot out there. So we know that almost 70% of US households own a pet. And that's 85 million households, and it works out to 94 million cats and 90 million dogs in the US. And this is as of the 2017. And $70 billion was spent by owners in 2017 on the care of their pets, huge amounts of money. And we know now that companion animals, so dogs, cats, and some other species, do develop many of the same diseases that are seen in people, like diabetes, heart disease, immune-mediated diseases, arthritis, many cancers, chronic kidney disease, et cetera. So they live in the same environment. They get many of the same conditions. And owners are increasingly interested in more advanced therapeutics for their pets and interested for novel approaches to treat their pet's disease. So why would we consider using companion animals as a model? Well, these diseases develop spontaneously, so they can take years to evolve. Our pets don't live as long as we do, obviously, but you can see cancer in the pet population emerging at age 6 to 8. We see chronic kidney disease, for example, in cats as they age 10 to 12 years. So these processes occur over time with age like they do in people. We do know that there's often a more relevant biology. So these conditions develop in an immunologically intact animal. And importantly, these aren't animals that are in a cage that have a really limited environment in which their immune system is sort of being educated. They're out and about, they're exposed to pathogens, their immune system's evolving with them. So much like you and I, they have an immune system that's diverse and has memory. The disease presentation is variable. So like humans, you see animals with the same disease in various stages or conditions when they present. And importantly, we do see response and resistance to standard therapeutics, which is important because many drugs and devices are developed in the setting of patients that have already failed something else. There's a higher incidence of some diseases, like certain cancers and immune-mediated diseases. The other thing that makes companion animal species, particularly dogs that are within certain breeds, interesting to work with is that they have something called long linkage disequilibrium, which means that within a breed, they tend to have large stretches of genetic information that are linked. So if you have a breed-specific disease, it makes it sometimes very easy to identify a genetic link because they share some genetic information. And then there are lots of opportunities for investigational therapies to establish pharmacokinetic and pharmacodynamic relationships. The other thing that we have as an advantage in veterinary medicine, which is slightly different than human medicine, is something called the species in kind approach, which is that for many devices and drugs that are being developed, dogs are a laboratory species that are used. So you actually are testing or evaluating initially a novel device or drug in a dog and then taking it to a pet dog. So you have the research dog and the pet dog. So that gives you a continuum of information within the same species that you don't have on the human side. We don't test these things in people before they move into people that are affected. So we have a lot of information typically that we've gathered in our healthy research dog before we move into the affected patient. And that allows us to really predict toxicities much more readily, to reach, for example, a maximum tolerated dose more quickly, and to really sort of predict what our pharmacokinetic and pharmacodynamic endpoints may be using data that we've generated in the laboratory dog and then putting that into an affected dog. And just to sort of give you an example of timelines, so we can typically get a phase 1 study in dogs of a new drug completed within 3 to 6 months. Because we have a lot of information in healthy dogs. In people, a phase 1 study can take years to get done. It's slowly enrolled, it plods along, any sort of flag of toxicity has to be evaluated. So it can take years to just sort of get through that initial process of vetting a drug in the humans, where we do it much more rapidly because we have that continuum. So what does the regulatory landscape look like in veterinary medicine when we think about some of these translational trials that we may want to perform of drugs that may enter the human realm? So clinical trials in companion animals don't require FDA approval or FDA monitoring at all unless you have a drug or device that is heading towards veterinary approval. And even in the device realm, much of the device development is excluded from regulatory oversight in veterinary medicine, which is kind of interesting and very different than the human landscape. So you can develop a device for veterinary medicine without really any regulatory oversight. But if you're just looking at a novel drug or device that you want to put into people, the FDA has no oversight. So that gives us a lot of sort of room to move, to adapt, to really be innovative without having a whole lot of regulatory burden on top of us. So the early phase 1 and 2 studies that we do in our veterinary patients are often performed without FDA input. We don't need to file paperwork prior to the initiation of a clinical trial unless we're planning on registering that drug for use in veterinary medicine. So if I get drug X from a company and they say, we want to bring this drug into treat patients with cardiac disease, but we want to test it in dogs with cardiac disease first, we really don't have any FDA oversight at all. It just makes things move much more quickly. The other thing is that we don't need what we call GMP product or good manufacturing product to put things into veterinary patients. We need a high standard of product, but we don't have to have things manufactured to that standard, and that cuts the cost quite a bit when you're moving new drugs into the veterinary realm for testing. We do have a standard called Veterinary Good Clinical Practice, much like Human Good Clinical Practice, but it's a guidance document, not a regulatory document. So that means that these are the guidelines that the FDA has provided in terms of good clinical practice, but you're not bound to adhere to them. Nevertheless, we do strive to adhere to those guidelines. And now, within the veterinary realm, clinical trials are becoming an integral part of veterinary medicine in both the specialty and private practice setting. And there are many things that are driving that. Pet owners want new things to treat their pets, but also many of these trials come at a reduced or absent cost so that it's free for owners. And in our profession, we're a cost driven profession, meaning there's really no health insurance or secondary or tertiary payer that helps with costs. And so people are paying out of pocket. And if they enter a clinical trial, usually they don't have to pay anything. And so that certainly helps with managing costs associated with medical care. So again, who benefits from this? Both humans and pets benefit from clinical trials. The pet owners have access to advanced state-of-the-art care, often at little to no cost, and then we get new information regarding a disease process and response to therapy that can be used to advance the treatment of human disease. Within the regulatory setting, in an academic setting, or even sometimes in private practice, there is some form of approval that's required. At most institutions that resides in the IACUC. Most veterinary institutions require an IACUC approval in order for a clinical trial in veterinary patients to move forward. There's no IRB or HIPAA regulations currently. And then at some academic veterinary centers, there's a hospital oversight committee that provides oversight of the studies. Sometimes they're involved in just approving the consent form, et cetera. And then informed consent is required for all trials in companion animals. And that's it's a guidance, but it's typically something that veterinary institutions adhere to. What would we expect veterinary trials to contribute to therapeutic development? One, we would want them to provide information regarding expected and unexpected adverse events, especially in the context of comorbidities. This is really important information. You want to move into your human population knowing what happens in a patient that may have some underlying kidney disease or heart disease. What happens in a patient that's already got some loss of quality of life. Understanding the effects and context of comorbidities is important. We would hope to establish an efficacious dose and regimen. Understanding how we dose the drug, not just from the actual physical dose, but do we give it every day? Do we give it twice a day? Is it given twice a week? And we've worked with several companies on this question. What's tolerable, what's effective, how can we mitigate those side effects by changing dose and regimen? The other thing that we potentially can contribute is evaluating novel drug combinations. These can be really challenging to test in people, particularly in patients that may not have received lots of prior care. So combining things in an interesting way in a landscape where we aren't burdened by the regulatory oversight makes it a little easier to move those things forward. Another big plus on the veterinary side is the opportunity to treat naive disease, and this really cannot be overstated. In human clinical trials, most patients have failed other regimens. There's some upfront standard of care that's required. So often by the time they get to a clinical trial, they may have exhausted multiple therapeutic regimens. And so then it's possible that a therapeutic that might have worked in a patient that had not been treated before is now going to fail because that patient has lots of other stuff going on, their disease is too far progressed, or they are simply the landscape of that disease has changed and that therapeutic is not going to work anymore. So we are not beholden to standard of cares because we don't have third-party payers that dictate reimbursement. So we can take groups of patients that have never been treated and enroll them in novel trials. And that gives us lots of information that's really important to the human landscape regarding how a drug may behave in a setting where a patient has never been treated. The other thing that we have the ability to do and that we do now routinely is longitudinal assessments of individual patients. So because our patients are not mice and you don't have to kill a mouse at each time point, we can collect biomarkers from them over the course of treatment. So we often do that. So we're collecting plasma, we may be taking biopsies, et cetera, and we're cataloging those in a longitudinal manner. We also have validated health-related quality of life assessments. And again, that's important. Even though our patients can't talk, they're much like pediatric patients. so their pet parents are able to assess their quality of life. And we have documents that they fill out at each visit, and that gives us a really good sense of, maybe the drug's working, but their quality of life stinks. And owners will say, I don't want to continue on this because my dog doesn't want to do any of the things that he wanted to do before. And that's an important flag for the human side, because if that's happening, then it's probably going to be worse on the human side as well. Importantly for you all, when you're considering what sort of models to use, the information that we gather in these preclinical trials can be included in IND and IDE applications. And I've been involved in many trials where we've put together a document that has provided either supplemental information or key components for an IND that has really helped a company move forward with confidence that the drug is going to be safe, that it hits its target, that it has a biologic activity. But from the FDA standpoint, they look upon this data in a favorable manner. So if they see that a spontaneous model of disease in the veterinary population has been involved in development, and that they see that it's been safe there, they see that it's met some critical endpoints, then it certainly helps them with confidence that they're not going to encounter random, unexpected adverse events on the human side. So this is critical information that can move the process forward. So I'm going to talk to you about some of the spontaneous disease models that you may not be familiar with that are used to help model human disease. So there's a number of canine cardiovascular diseases that are out there that actually have significant similarity to the human condition. So there's something called arrhythmogenic right ventricular cardiomyopathy, or ARVC, which occurs in boxer dogs and some other dogs, but it's really prevalent in that breed. It has a striking similarity to the human condition, and we know now that both are driven by specific mutations that affect cardiac myocytes. So ARVC is a really challenging and can be fatal condition on the human side, and it's fatal on the veterinary side as well. So this is a great model that's spontaneous, occurs in these dogs as they age, that really mimics the human condition and can be used to test a potentially new therapeutic to treat ARVC. Chemotherapy-induced cardiomyopathy. So doxorubicin is a chemotherapy drug that's used in human medicine and veterinary medicine. And one of the challenges that we're facing now in human cancer treatment is that many patients are living a long period of time and developing chronic toxicities 10, 20 years down the line. It's particularly relevant for kids in disease entities where they're cured, but then 20 years down the line they've got issues. And one of the big ones is heart toxicity from doxorubicin, which is one of the most common chemotherapeutics used. And we see doxorubicin cardiomyopathy in dogs, and we have formed a cardio-oncology working group to identify early biomarkers of cardiotoxicity in dogs and then have this validated in people, so that we can potentially pick cardiac damage up earlier than we do now. It requires imaging that isn't ideal, and identify those patients that are more likely to develop essentially clinical toxicities down the line. Small molecule inhibitors that are used in cancer therapy often induce significant hypertension in people, and so it affects vascular biology. We see that in dogs as well. There are many spontaneous cardiomyopathies, like dilated cardiomyopathy in Dobermans, which serve as a good model for the human disease, and mitral valve endocardiosis, which is actually a relatively uncommon condition in people, affects about 2% of the population of cardiac patients, but it's prevalent in, for example, King Charles Cavalier. So they're a great model of mitral valve endocardiosis. This slide is courtesy of Dr. Joan Coates at Missouri, but it shows you the landscape of spontaneous canine neurodegenerative diseases, where multiple mutations have been identified in multiple neurodegenerative diseases that really mimic what's seen on the human side. So there's a whole landscape of neurodegenerative diseases that you could evaluate novel therapeutics for in dogs and then potentially translate that into people. And one of the ones that's been a focus for a while is SOD1 mutant ALS. And so SOD1 mutations are actually fairly common in a condition called canine degenerative myelopathy. And there's an ongoing effort to target SOD1 inside the central nervous system using basically AAV-associated vectors to essentially downregulate SOD1 expression. So this is actually a pretty good model of the human disease in a condition where essentially no therapeutic advances have occurred for decades. There are many respiratory diseases that occur in veterinary patients that have human correlates. For example, idiopathic pulmonary fibrosis occurs in West Highland White Terriers. We don't really understand why they have such a prevalence of this disease. It has a similarity with a human disease with respect to CT findings, clinical disease sign progression, and like the human condition, it's refractory to most treatments. And so it's a great model for really evaluating novel therapies for IPF in people. And then I bet you didn't know that cats are the only species other than people that get spontaneous asthma. So asthma is very challenging to study in other species because you have to induce it. Mice don't get it naturally, but cats do. And so it's been a well-characterized model of asthma. It has clinical characteristics that are seen in human asthma, eosinophilic airway inflammation, airway hyperactivity, and many of the same CT changes. So one of my colleagues has worked on this disease in cats for a long time to help develop novel therapeutics and has tested some novel targeted agents. Another interesting disease that has a really direct correlation with a human disease, they're almost identical in terms of how they present, is something called atopic dermatitis, which is actually increasing in the human population. It's essentially an inflammatory disease of the skin that's an immune dysregulation. These are sort of the features that are shared between humans and dogs, and actually it looks very similar on clinical presentation. And we know now that this disease, which is driven by many things, but also by significant itch, really sort of this need to scratch in both species, is driven by a cytokine called IL-31, which creates this intense pruritus. And actually on the veterinary side, we now have 2 targeted therapeutics for this, and we were actually ahead of the human side on this one. So we have an antibody that targets IL-31 and markedly inhibits that pruritus. But it's a great model of the human disease to develop new therapeutics, and anti-IL-31 therapies are now on their way based on the success of what happened in the veterinary side. So I'm just going to focus now for the rest of the talk mainly on canine cancer, because that's really where I work. And I'm going to give you sort of a vignette of where we sort of moved from in vitro to normal dogs to affected dogs, and then how this translated back to the human side. So cancer is extremely common in the dog population. If you look at overall diagnosis, and this is back in 2011, it's probably higher now, 4.5 million tumor diagnoses per year. 1.5 million of those are cancer, new cancer diagnoses in the veterinary population. When we look at the actual age-adjusted cancer incidence in dogs, it's higher than it is in people. So we see it a little bit more frequently than we do in the human population. We look at the spectrum of disease. Some of them are different, some of them are similar. We see a lot of bladder cancer, for example, a lot of lymphoma, carcinomas, bone cancer, soft tissue sarcomas, melanomas, et cetera. So many of the diseases or cancers really overlap with our human counterparts. So, why would we want to study cancer as a model? Well, it turns out when you look at how DNA is organized in the chromosome, in dogs, it's much more similar to in people than it is in rodents. So when we look for dysregulation of chunks of DNA, it's easier to find those links in dogs and humans versus mice and humans. We also know that there are similar gene expression alterations, similar targets have been identified, and drivers of tumorigenesis. More importantly, cancer that we see in the pet population recapitulates what's seen in the human population. It's heterogeneous, it recurs, it metastasizes, and resistance to therapy is common. Again, we don't need GMP quality material to do a lot of our clinical trials, and that pre-IND work in our veterinary cancer patients has turned out to be of significant value during the drug development process. Our patients, again, are less heavily pretreated, and that makes a difference when we're evaluating a new therapeutic to really get a sense of what it would look like in a patient that hadn't failed multiple therapies. And lastly, we use similar imaging and treatment modalities in the veterinary population. So we use CT scans, we use MRs, we use PET CT, all sorts of advanced imaging and advanced treatments. So we also have linear accelerators we use for radiation. We have a whole slew of immuno-oncology reagents that are coming our way. So the tools are really becoming very similar, so it makes it easier to sort of recapitulate in our patient population in veterinary medicine what you might do in the human population. And the NCI has now really decided that this is important, particularly in the landscape of immuno-oncology. I'm sure many of you are familiar with the sort of massive successes we've had with some of the checkpoint inhibitors, like Keytruda, which is always on the news. But for all those patients that have been sort of home runs with respect to immuno-oncology therapeutics, the overwhelming majority of patients still fail to respond to immuno-oncology agents. And we don't really know why. And it's been very challenging to sort of figure that out in the mouse models. I think we're getting there very slowly, but the NCI has now, using moonshot efforts, decided to invest in dogs as a model of cancer in the immuno-oncology realm. And the first thing they did was to really sort of put a bunch of money into addressing one of the gaps we had, which is understanding the canine tumor genome. So there's a huge effort now. We've sequenced many of the canine tumor genomes. There are many places where these genomes overlap. So we just finished doing a whole slew of work with canine bone cancer, osteosarcoma, and it really does mimic at the genomic level the human disease. And so this sort of information is sort of building the tools in the toolbox to provide a better model for the human condition. And then these are some instances where we've worked together with pharmaceutical companies to really help advance their drug development pipeline. And I'm going to focus on this story in the end. But this was one of the agents that we worked with years ago, sunitinib, also called Sutent. It has a cousin called tosarinib, and that's the drug that we physically worked with. We did this clinical work back in 2000 to 2001, showing that the drug was tolerable, it had biologic activity, and it targeted a receptor called KIT. And we established PK-PD relationships, and this facilitated approval of sunitinib in people for something called gastrointestinal stromal tumors. So the data that we generated went into the IND and then facilitated that drug development path. It also helped choose where to place that drug in the human patient spectrum. Another drug that we worked with is called an XPO1 inhibitor. The human version is selinexor. The veterinary version is verdinexor. We use dogs with lymphoma to validate target, pharmacokinetic and pharmacodynamic relationships, tolerability, biologic activity, and now verdinexor is being developed for veterinary patients. Importantly for selinexor, the clinical trials that we did in dogs established the dose, the regimen, and how to mitigate toxicities, which is loss of appetite. So that laid the groundwork for the human clinical trials, and this drug has just met its phase 2b endpoint and will get approval for multiply resistant myeloma. And it actually may be much more effective in the upfront setting. Telasalib is a PI3 kinase gamma delta inhibitor, and we used a very similar drug in dogs with lymphoma to validate target, PK-PD relationships, tolerability, but importantly, activity in peripheral T-cell lymphoma, which hadn't been necessarily flagged, but we had most of our activity in the T-cell lymphoma realm, and that's where the human clinical trials were placed, and they've just been fast-tracked for approval in the setting of T-cell lymphoma in dogs. And then I'm going to talk about this last one here. So I've worked on a receptor called KIT for many, many years, really since 1994, which is a receptor tyrosine kinase. It's important in the development and growth of many normal cells like melanocytes, mast cells, et cetera. It has a ligand called stem cell factor. It's on the cell surface, and it's a typical receptor tyrosine kinase. We identified several years ago that mast cell tumors in dogs carry activating KIT mutations in this part of KIT that turn the receptor on constitutively, and that these mutations look identical to the human, the mutations that are found in KIT in human GIST or gastrointestinal stromal tumors, and that these KIT mutations were associated with the high risk of recurrence, metastasis, and death, and that it was an oncogenic driver of disease. So dog mast cell tumors were a great model of KIT dysregulation because about 30% of them carry these mutations. We know their biology, and it has a human correlate. In the GIST realm, over 50% of patients have those mutations. And again, 30% of the dogs have them. And so if you look at sort of the spectrum of mutations that are identified in dog mutations, they're here, and they overlap with the human mutations that are found. And again, these mutations turn on the receptor. So we were approached by a company that was interested in developing a novel KIT-targeted antibody. There are several small molecule inhibitors of KIT that have demonstrated good activity, but there was an interest in sort of developing an immuno-oncology agent to target KIT that would circumvent some of the disease or drug resistance mechanisms associated with the small molecule inhibitor. And so the company was interested in working with us because they had this humanized anti-KIT, but the clinical trial of another anti-KIT antibody had been halted due to severe anaphylaxis secondary to the antibody administration that was not predicted from their mouse models and not predicted from the non-human primate models. So they were 3 patients in, all of them had anaphylaxis. None of the preclinical modeling predicted that. And so they came to us for a couple of reasons. One, dogs love to anaphylact basically to anything. So their mast cells are hypersensitive, and so they tend to degranulate when you look at them wrong. So if you cleared the hurdle of anaphylaxis in dogs, you'd probably clear it in people. The other reason was that the antibody that they had generated did not bind rodent kits. So they couldn't even use it to test mast cell degranulation in rodents because it didn't bind rodent kits. But it did bind non-human primate, canine, and feline kit with very high affinity. And so they came to us to really say, can we test this antibody in dogs to determine, well, is it going to cause mast cell degranulation? Does it bind KIT? What does it do? Does it have biologic activity? And can we use your mast cell tumor model to really define some of those parameters? So this is the antibody. It blocks essentially the dimerization by binding in this region. And they were able to show that it's actually much more potent at inhibiting proliferation of KIT-driven cells than the small molecule inhibitors, so much more potent. And so it looked good based on in vitro studies. And then they could show that it actually does inhibit phosphorylation of KIT in vitro. So here's KIT phosphorylation induced by dog stem cell factor, and you can block it with the antibody, and you can block it with a small molecule inhibitor. So it had in vitro activity. And then we took this one step forward and put it into normal, healthy research dogs. So here's our transition into the species in kind. And we asked a couple questions. Is it safe? Do the dogs anaphylax? And what does it do to the normal mast cell population? So here you can see that we took normal dogs, gave them doses of antibody. They either got 10 or 30 mg per kg, and then we counted the number of mast cells that were left in their skin using skin biopsies to see if we depleted the mast cell population. And you can see that within 7 days, most of the mast cells in the skin were gone, and that at the higher dose, they stayed suppressed for a longer period of time. So we knew that we were hitting a target. We also knew that these dogs really didn't have any toxicities that we identified. So they cleared the anaphylaxis hurdle. So that led us to a clinical trial where we took dogs with mast cell tumors that we know have KIT on their surface, often have KIT mutations, and elected to try a couple different dose levels and schedules of this antibody. Now remember, this is a humanized monoclonal antibody that's being given to a dog, so you have a limited number of cycles that you can give it before they develop anti-human antibodies. So we did serial tumor biopsies, lots of PK/PD, and weekly assessments. So this is where we can actually multiply sample and longitudinally follow a patient to gather biomarker data about how that drug is working. And lo and behold, we actually were surprised that we had substantial responses to therapy. And we had a couple different groups that we tested. So lots of dogs with just even a single dose had significant reduction in their disease burden. Some of these actually had mutations and responded as well. And you can see if this was baseline KIT phosphorylation, most of the patients actually exhibited downregulation of KIT phosphorylation following antibody administration. So we had basically an indication that there was biologic activity. We had an indication that we're hitting target. And we actually could show significant clinical activity in patients that had failed disease. So this is a multiply failed patient who had a huge mast cell tumor, failed surgery, failed chemotherapy, failed radiation therapy, failed another small molecule inhibitor, and responded to therapy. So this mimics the human condition. You've got a multiply failed patient who has a response to therapy. And again, that gives you a sense that what you're going to see in people is likely to be concordant with what you see in the pet population. We actually had a lot of patients that, based on our typical RECIST criteria, we measured the tumor, we thought still had tumor left, but when we took them to surgery, histopathologically, it was just fibrosis. So we had some patients that actually had no disease left, even though they were partial responders at histopathology, they had no disease. And we actually had clearance of some metastatic lymph nodes, which was quite phenomenal. So more activity than we actually thought we were going to get. We were able to look at the pharmacokinetics of the antibody in vivo. So this is, and it was dose proportional. So here's your 10 mg per kg and then your 30 mg per kg dose. We were also able to show that we actually developed KTN-0158 antibodies, so neutralizing antibodies over time, and that actually affected the pharmacokinetic profile. So we had decreased efficacy on the second round because we had antibodies that neutralized our product. But this was sort of an example of the sorts of clinical trials that we try to do, where we tie in vitro information to information generated in healthy dogs and then moving them into dogs that have cancer. And then this gets put together in a packet like this did in an actual technical report that went into the FDA. And this was really important for this company, primarily because the previous antibody, while they were developing their antibody, the previous antibody was shown to cause anaphylaxis. So the FDA was really, really nervous about putting another anti-KIT antibody into people, and this helped give them confidence that not only would it be safe, but that they would see biologic activity. So the first human patients were safely dosed, and this is an example of how long it takes on the human side. This study's been going on for 2 years now. They're still in their phase 1 setting, so it's taking forever to sort of dose escalate, where we finished this thing up in a couple months. So what are the sources for comparative oncology out there? And I'm going to go through some of the resources that you might want to tap into if you're thinking about potentially using spontaneous models. So within the oncology realm, there's actually a comparative oncology program that's housed in the Center for Cancer Research at the NIH, NCI. So this has been there for over 10 years now. And it's really driven to characterize naturally occurring cancers that develop in pet animals as a tool to study the human disease. So it's embedded within the intramural NIH. This comparative oncology program leads something called the Comparative Oncology Trials Consortium, which is a network of now 25 academic centers that run comparative oncology trials. So it's essentially a huge trial network that will execute many complicated studies that are often in collaboration with pharmaceutical industry, academia, not-for-profits. And many of these trials involve multiple PK endpoints, sampling, et cetera. And so we've run over 25 trials now, and many of these have really helped to drive human projects forward. Another new initiative out of the comparative oncology program is the Comparative Brain Tumor Consortium, which now brings together a whole group of people from the extramural community, the NIH, NCI, and puts them together to really characterize many of the brain tumor models that are seen in dogs, to define new drugs and new therapeutic targets, to perform clinical trials, and to follow patient outcomes. And as you know, glioblastoma, as I mentioned before, is really a devastating disease on the human side for which there's been very little progress. Dogs get very similar forms of glioblastoma, and there's actually 2 very large immuno-oncology clinical trials in dogs with glioblastoma now, with the hope that some of those therapeutics that are being developed will be successful and move on to the human side. Another effort that is growing quite rapidly now, and one of which we're a part, is called the CTSA One Health Alliance. And so this is comprised of now 13 veterinary schools partnered with medical and other colleagues through a National Institute of Health Clinical Translational Science Award. So we have a CTSA award here at Ohio State, and all the other institutional partners do as well. So if you're a vet school with a CTSA or a CTSA partner, then you can be part of this network. And this really is sort of a broader mission to advance understanding of diseases shared by humans and animals. And it leverages expertise across really a broad spectrum of clinicians and scientists to really connect, whenever we can, human and animal health and learn from both. So the COHA community is growing rapidly, and so we're now connected in a very fundamental way, and we've implemented standardized processes across this community. So this is now 13 academic centers that are following many of the same practices, and that's including good clinical practice. We've developed standard operating procedures for trial evaluation, approval, client consenting, and monitoring. We've implemented REDCap across the network. Now most people are actually using REDCap as their electronic data capture tool, which will make it much easier to synthesize data across institutions. We're developing clinician scientist education support through these fellowship programs, and so that's a relatively new module that's being developed. We have pilot grants that have been funded that have really helped move this effort forward. And one of the larger things that's occurring right now is the connection of veterinary electronic health records. So we've migrated to an electronic health record platform in veterinary medicine. You guys are probably all familiar with Epic, which is one of the larger ones that's used on the human side. Ours is a bit more fragmented, but we've developed a platform now to integrate all of those and to really allow us to do cohort discovery across a large geography, which is something we haven't been able to do. So this connection will really create a tool for researchers as they move forward to leverage some of these spontaneous models. We have a number of subcommittees within COHA that are building these tools. We have a virtual biospecimen repository which we're developing. This is really important because there are a number of biospecimen repositories around the country that are not connected. And if you're a researcher and you say, well, I want to grab serum from 20 patients that have ARVC and box or ARVC, then you can potentially log in through a virtual biospecimen repository, find out where those samples are, and get access to them. So that's something that we're planning on building. We're also building a curation of all the spontaneous disease models as they evolve, so that we'll be able to have that data on our portal, which tells you, okay, what's this disease entity? Well, does it have the genetic mutation I'm looking for? What is the clinical presentation? How is this treated on the veterinary side? How many patients are potentially available per year for a clinical trial? So those sorts of tools are really helpful. These fellowship training awards, and then the connection of the human and veterinary OMOP platforms, which is, can we not only connect our EHRs within veterinary medicine, but can we then connect them back to human medicine to find commonalities and do cross-species research? Within this institution, we actually have a pretty phenomenal shared resource that's developed over the past 10 to 15 years. It's now a shared resource associated with a cancer center, and both the CCTS and the Cancer Center were instrumental in helping build this and support this. So we have a clinical trials office and shared resource that really is meant to advance the diagnosis and treatment of diseases in veterinary patients, but also impact human health. And we now have 8 full-time staff. It's much like a human office, human clinical trials office, with nurses and administrative support to facilitate clinical trial endeavors. It sort of has 2 main components: the Blue Buffalo Veterinary Clinical Trials Office, which really embodies the functionalities associated with clinical trials, and then the biospecimen repository. So we have our own in-house biospecimen repository that is now being leveraged by not only inside entities, but lots of pharma is now querying this biospecimen repository, and also other academic entities. And then one of the other resources that you may not be familiar with is the Translational Therapeutics Think Tank, or T4, which is sort of a body of people that works to help investigators early on in their preclinical studies, to help them identify what their gaps may be, address any roadblocks, find appropriate collaborators and partners to facilitate their work. And we meet monthly to do this, and every time we meet, there's something new comes out of this. We find a new collaborator, we help them solve a problem. So this is sort of an incubator that is something that you may find useful for you as you think about animal modeling. So to summarize, I'm very passionate, obviously, about including basically spontaneous disease models in our veterinary patients in the development of new therapeutic approaches. I think there's no question that there are multiple benefits to this for our pets, the pet parents, and also the human enterprise, so that we can really affect outcomes on both sides of the aisle, and hopefully markedly reduce some of those failure rates, which really are not sustainable long term. And if we were to think about what this could do, we could potentially, by just removing some of the drugs out of the pipeline early on before they hit some of those clinical trials in humans, save billions of dollars that could be reinvested in other efforts. And this just sort of models what would happen if you did just occasionally take a few drugs out of the pipeline. You'd have less migrating through into that later stage study, and that's the money that you would save. So again, none of this work ever happens without a whole team of people, and I've been fortunate enough to be parts of really phenomenal teams that have really helped move the paradigm shift more rapidly than I would have thought possible in the past 5 years. And so I'll stop and be happy to answer any questions. Yes. Sure.
Everything will be listed on the CCTS website.
Do you have a question, Chris?
Yeah, great talk, Cheryl.
Thanks.
I was just fascinated with this approach, and I'm always surprised, we're working in obviously a lot of variety of drugs and device development, and the scenario is still so unique to me. And so one thing is, it's surprising how often the oversight's not really required from the FDA's perspective. Do you ever feel that, is there a pushback from IARC on bringing some of these novel programs in? And do you ever engage FDA preliminarily before initiating this?
So with respect to device development, it is extremely odd that the Center for Veterinary Medicine at the FDA is not at all interested in overseeing device development in veterinary medicine, because that has created a niche where companies want to say something's a device so they don't have to go through any regulatory processes. With respect to the pushback from IACUC and other entities, yes, we sometimes get pushback moving things into our patient population, much like you would want a set of safety data that's required before moving into kids. So they do require some level of evidence that the patient population is going to be safe with whatever dose or agent you're going to use. The FDA, the Center for Veterinary Medicine, the FDA, does not provide comment on almost anything. So when we actually asked them for comment on our GCP module, and my contact there gave me comment and then said, oh, I shouldn't have given you comment because I'm not supposed to be doing that. So they really try to stay out of that process. And we haven't actually interfaced with the human side early on. Although they have been involved in many of our sort of national meetings. So the FDA has made it really clear that contrary to what people think, adverse events that occur in our veterinary population alone would not be enough to derail a drug or device development. They take the totality of the data and decide whether or not to move something forward. Pharma has been very reluctant sometimes to use these models because they say, well, what if something bad happens? And the FDA is not going to let us move this forward. And the FDA has very clearly said that's not the case. If something's going to die, it's going to die because it was going to die, not because one dog had an adverse event. And so that's really helped, particularly with the smaller companies, take the risk, which they've often found to be an acceptable risk. In the yeah, so they've had some stable disease, but they're so early and they're pushing that dose forward. And they've made some modifications to the antibody that change the FC receptor a bit, so that it doesn't bind at all. And so they went back and changed which made them have to restart things. But I've actually been in contact with them because it was bought by a company called Celldex. After this data came out, Celldex picked them up, and so they're still moving. It's just taking a long time. Yes.
So it's about mice model, using mice model for even though we know that they are going to fail in clinical trials, I'm surprised why in every and each grant and papers we have those experiments. So do you see in a few years that it will grow and
So mouse models are extraordinarily useful as long as you take the data in its context. So I think mouse models are very useful to say, yes, the drug hit its target. Yes, if the target's there, the tumor will shrink. Yes, we can induce an immune response. I think the problem has been not so much the mouse models, it's how we interpret them. There's no direct line to people. So you can't say just because it worked here that it's going to work there. And I think that's been sort of the overinterpretation, is that we often tend to jump very rapidly based on early successes in mice into people without thinking about what are the mitigating factors in the human side that may influence response. Mouse models are phenomenal. I use mouse models, but they're a tool, and they're one piece of the puzzle. You can't make them the only piece of the puzzle. Okay, thank you.